Welcome back to the SANS Channel podcast, where we discuss the coolest scientific papers and preprints of our day.
Great to be back.
If you're enjoying getting the deep dive on these fascinating topics, please do like the video and subscribe. It really helps the channel grow.
Absolutely. And today, we're looking at a really intriguing new idea about why we sleep.
Yeah, something that goes way beyond just, feeling rested or memory consolidation.
Exactly. Forget the usual explanations, just for a moment.
Because we got this paper right. It suggests a completely unexpected role for something we thought we basically understood. Myelin.
The insulation around our nerve fibers, yeah.
The paper's titled Myelin, a possible proton capacitor for energy storage during sleep and energy supply during wakefulness. That title kind of gives it away, doesn't it?
It certainly does. It hints that myelin might be acting essentially like a battery for our brains.
A battery? That's quite a concept.
It is. This research proposes that myelin, which, we typically just think of as speeding up nerve signals.
Right, just insulation.
Might also be absolutely crucial for how our brain stores and then uses energy.
So the idea is, during sleep, it's charging up somehow.
That's the proposal. Charging during sleep, and then during wakefulness, it helps power our brain activity.
Wow. Like a built-in backup power supply right there in the wiring.
Sort of, yeah.
So for this deep dive, we're really focusing on this one scientific article. And it pulls together quite a few different threads, doesn't it?
It does. It looks at how myelin behaves metabolically, the surprising discovery of energy-producing bits within myelin.
Which we'll definitely get into.
And even how things like sleep patterns and the amount of white matter, which is mostly myelin, differ across various animals.
Okay, so our mission today is to really unpack this proton capacitor idea, see what the evidence actually suggests.
Let's dive in. So maybe we start with the big picture. Brain energy and sleep. Why do we even need to sleep for energy?
Well, we know some reasons for sleep, right? Tissue repair, memory stuff, clearing out waste.
The lymphatic system, yeah, all important. But the primary reason, the main driver, has always felt a bit elusive.
Agreed. And a leading hypothesis has always been that sleep, fundamentally, lets the brain replenish its energy reserves.
Exactly. And for a while, scientists thought the main reserve was glycogen, stored sugar.
But the brain doesn't actually store that much glycogen, does it?
No, not really. And realizing that kind of shifted the focus towards mitochondria, the cell's power plants.
Okay, makes sense. But where does myelin fit into this energy picture?
Well, this is where it gets interesting. It turns out sleep isn't just downtime for neurons. Things are happening in myelin, too.
Active changes.
Yeah, studies have shown changes in gene activity related to making and maintaining myelin during sleep. Even the physical structure seems dynamic.
So it's not just passive insulation sitting there?
Apparently not. And this paper suggests these sleep-related changes aren't just a side effect of neurons resting. The idea is that sleep might actively maintain the health and maybe even the energy-storing capacity of our white matter.
And we also know ATP levels, the main energy currency, can fluctuate in neurons during sleep cycles.
Right. So you've got myelin changing during sleep, and sleep clearly linked to brain energy management.
Now here's the twist you mentioned earlier. Myelin isn't inert. It actually contains components that look and act like tiny bits of mitochondria.
Yes, this was a pretty surprising finding in recent years. These components can actually produce ATP.
Generate energy inside the myelin sheath.
Using oxidative phosphorylation, or OXPHOS. It's the main, highly efficient energy-producing process in our cells. So yes, like tiny power plants embedded within the insulation.
Okay, hold on. Where do these mini power plants come from? They're not full mitochondria, right?
No, the likely source is something called mitochondrial derived vesicles, MDVs.
Vesicles, like little packages.
Exactly. Think of them as tiny delivery packages sent out from the main mitochondria in the neuron, carrying essential machinery to the myelin sheath.
And these MDVs can actually make ATP on their own once they're in the myelin.
That's what the research suggests. They can contain a functional ATP synthase, that's the enzyme that actually builds ATP, and operate independently.
Wow. Okay, so that really sets the stage for the main hypothesis of this paper.
The proton capacitor hypothesis, yeah.
Lay it on us. What's the core idea?
So the central idea is that myelin acts like a capacitor, but for protons, positively charged hydrogen ions.
Storing charge.
Essentially, yes. Storing energy in the form of a proton gradient. The idea is, during sleep, myelin actively accumulates these protons.
Charging up.
Charging up. And then during wakefulness, it releases them in a controlled way to drive ATP production via that RxPHOS machinery we just mentioned.
Has this idea been floated before?
The general concept of proton capacitance has, but this paper really tries to flesh out a specific biological mechanism involving myelin proteins.
And they even did some calculations, right, about how many protons could be stored by the key myelin protein.
They did. They looked at proteolipid protein, PLP, and myelin basic protein, MBP, based on rough calculations of how many protons these proteins could potentially bind.
Okay.
They estimated that a typical human sleep duration, say around 8 hours, might actually be the time needed for this myelin recharging to happen sufficiently.
8 hours to charge the brain to wiring. That's quite specific.
It's an estimate, of course, but it connects the hypothesis to observable behavior.
Okay, so myelin as an energy store. Let's quickly touch on its structure. Formed by oligodendrocytes in the brain and spinal cord.
And Schwann cells in the peripheral nerves. Yeah, layers wrapped around the axon.
And it's not solid plastic wrap, right? There's water in there. And its interaction with protons is actually how we can image myelin using MRI.
Precisely. That interaction is key for imaging, and maybe key for this function too.
So let's zoom in on those two proteins, MBP and PLP. How are they supposed to work together in this capacitor model?
Right. The hypothesis really depends on their specific structures and where they sit in the myelin layers. MBP first. It has this sort of flexible U-shaped structure.
Okay.
And it's rich in positively charged amino acids like lysine and arginine. The idea is that MBP acts like a proton buffer during sleep.
Like a sponge for protons.
Pretty much, yeah. Soaking up protons generated by the electron transport chain activity that's part of OXPHOS during the recharge phase.
Okay, so MBP absorbs the charge during sleep. What about PLP?
PLP is proposed to be the proton conductor during wakefulness.
The pathway out.
Exactly. It helps move those stored protons from MBP towards the ATP synthase machinery, specifically the part called the faux rotor, which spins using proton flow to make ATP.
And PLP has structural features for that.
It does. It has a high percentage of amino acids like serine and threonine, which are known to form efficient proton wires, pathways for protons to move rapidly.
And PLP spans the membrane, connecting different layers.
Yes, it has domains and different compact layers of the myelin sheath connected by this proton-conducting part within the membrane itself. It looks like a purpose-built pathway.
So the authors are suggesting a kind of closed loop for protons, involving these proteins and the membrane lipids.
That's the model. A localized circuit at what's called the major dense line involving the respiratory components, MBP, PLP, and the surrounding lipids, all working together to manage these protons.
This idea of significant energy production happening outside proper mitochondria, right there in the myelin, that's still pretty novel, isn't it?
It is, relatively speaking, but the evidence is building. Studies are showing ATP synthesis happening in myelin preparations from both the central and peripheral nervous systems.
And the key is that it doesn't necessarily need a big watery space for the proton gradient, like in mitochondria.
That seems to be the thinking. The respiratory complexes might generate proton movements more directly within the lipid-rich environment of the myelin membrane itself, a more contained charge transfer.
So can you walk us through the two proposed phases again? Sleep versus wakefulness.
Sure. So phase 1, during sleep. That's the redox coupling or recharging phase. The electron transport chain, ETC, is active, using NADH and FADH2, reducing oxygen and pumping protons, which then get buffered by MBP and PLP.
Charging the capacitor.
Exactly. Phase 2, during wakefulness, that's the proton-ATP synthase coupling or discharging phase. The stored protons flow from MBP-PLP through PLP to the ATP synthase, driving ATP production.
Discharging the capacitor to make usable energy.
Precisely.
And is there direct experimental evidence that myelin can sort of hold on to an energy charge like this?
There is some, yeah. Studies using isolated myelin fragments, sometimes called myelin vesicles, show that if you expose them to NADH...
The fuel for the electron transport chain.
Right. They seem to retain some kind of energy potential from that exposure. Because later, if you add the building blocks for ATP, they can actually synthesize ATP.
Using the stored potential.
That's the interpretation. And importantly, the longer the initial exposure to NADH, the more ATP they could make later, which really fits this idea of a charging mechanism.
Interesting. Let's go back to PLP, the conductor. You said it's the most abundant protein in central nervous system myelin.
By far, yes. And it's known to bind specifically to MBP, forming a complex.
Which makes sense if they work together.
It does, and its structure, those 4 transmembrane helices, the placement of charged residues in specific domains, it all seems geared towards this proposed role, especially those serine and threonine residues in the transmembrane parts, acting as the proton wires.
So during wakefulness, protons flow from MVP, somehow get onto PLP, travel through its transmembrane part,
Likely to the intraperiod line domain of PLP.
and they get funneled to the ATP synthase.
That's the proposed route. And intriguingly, PLP has actually been found to co-purify with parts of the ATP synthase complex,
Suggesting they might physically interact.
It certainly suggests a close functional relationship, and it's also worth noting, perhaps relevantly, that genetic defects in PLP are often linked with mitochondrial dysfunction.
Hinting at this interconnectedness between myelin integrity and energy metabolism.
Exactly.
Now, you also mentioned gap junctions earlier. Myelin is full of them. How do they fit into this story?
Right, gap junctions are channels that connect the layers of myelin and also connect myelin to the underlying axon. They're thought to control the flow of small molecules, potentially including ATP, into the axon.
Okay, so they're like gates.
Precisely. And the paper brings in this molecule called Oleamide, or OA.
OA, I've heard of that. It's known to induce sleep, right?
It is, and it's also known to block gap junctions.
Okay, I see where this is going.
The proposed mechanism is, during sleep, maybe increased OA levels close these gap junctions.
Preventing ATP from flowing from the myelin into the axon.
Exactly, which would help the myelin retain its energy and allow the proton accumulation, the recharging, to happen more effectively in MVP and PLP.
And then during wakefulness.
OA levels would presumably decrease, the gap junctions would open, and the ATP being generated by the discharging myelin capacitor could then flow into the axon to support its higher energy needs.
Is there evidence OA affects myelin ATP production?
Yes, the paper mentions experimental data showing that OA can actually inhibit ATP formation by those myelin vesicle preparations we talked about.
Consistent with blocking the output or maybe even influencing the charging?
It seems consistent with the model. And there's another interesting link, cytochrome C.
Key part of the electron transport chain?
Right. Cytochrome C can apparently catalyze the formation of OA.
Seriously? So high energy production might lead to making more OA.
That's a potential feedback loop, they suggest. Active OXPHOS during wakefulness could generate OA, which then helps block ATP outflow and primes the system for the sleep recharge phase.
That's a neat potential regulatory mechanism.
It is.
Did the paper try to put any hard numbers on this proton storage idea? You mentioned the 8-hour estimate.
They did attempt some quantitative estimates, yes. Very approximate, of course.
Ballpark figures.
Exactly. They estimated how many protons might be moved by the myelin's metabolic activity over, say, 8 hours of sleep in a cubic centimeter of myelin. And they came up with a number around 16 times 10 to the 20 protons.
That's a lot of protons.
It is. Then they estimated the total proton buffering capacity of that same volume of myelin, considering MVP, PLP, and also the phospholipids in the membranes.
And what did they get for the capacity?
Their estimate was around 10 times 10 to the 20 protons.
So the number of protons potentially moved during sleep and the estimated storage capacity are actually in the same ballpark.
Roughly, yes. Same order of magnitude. Which doesn't prove the hypothesis, of course, but it suggests it's at least quantitatively plausible. The numbers aren't wildly off.
It provides some initial grounding for the idea.
Exactly.
Now, the paper also took a broader view, looking across different species. Something about white matter and sleep duration.
Yes, this was quite interesting. They looked at data from 23 different mammal species and found a positive correlation between the total volume of white matter in the brain and the average total sleep time for that species.
So, generally speaking, animals with more white matter tend to sleep longer.
That's what the correlation suggests.
And since white matter is mostly myelinated axons.
They propose this correlation might really reflect a link between myelin content and sleep time.
How do they interpret that link? Why would more myelin mean more sleep?
Well, one interpretation they offer is that maybe having more myelin allows for or necessitates higher levels of neuronal activity during wakefulness. More complex processing, perhaps.
Which would mean a greater energy demand.
Exactly, leading to a greater need for the myelin energy system to recharge during a longer sleep period.
And conversely, less myelin might mean lower energy demands during wakefulness and thus less sleep needed for recharging.
That's the potential implication, yeah.
Did they relate this to humans specifically?
They touched on it. They mentioned the pattern of sleep duration across the human lifespan, how it's typically shortest in middle age.
Right, when white matter volume is often thought to peak.
Exactly. That inverse relationship might fit the pattern. And they also noted the general observation that women, on average, tend to have slightly less white matter than men.
They often report needing slightly more sleep.
Correct. Again, it's just a correlation, lots of potential factors, but it's another piece that seems potentially consistent with this idea of a link between myelin quantity and sleep need.
Okay, so let's try to pull this all together. What's the big takeaway here?
Well, I think this deep dive really highlights a potentially fascinating and much more active role for myelin than we usually consider.
Definitely beyond just passive insulation.
Right. This idea of myelin as a dynamic proton capacitor, actively involved in storing energy during sleep and supplying it during wakefulness. It offers a really fresh perspective on why we need to sleep. It connects sleep need directly to the brain's wiring and energy management.
And that cross-species correlation between white matter and sleep time is pretty provocative, isn't it?
It really is. It raises some profound questions about how brain structure, energy demands, and sleep habits are all intertwined across evolution.
And the potential role for Oleamide as a kind of natural sleep regulator that specifically interacts with this myelin energy system, that's also really intriguing.
Yeah, lots of potential angles there.
So this research clearly opens up a lot of new questions, new avenues for future work.
Absolutely. Could we eventually target myelin metabolism, maybe Oleamide signaling, to help regulate sleep? How does this proposed system fit with, all the other things we know, regulate sleep circadian rhythms, adenosine, all that?
Lots to explore. And for you, the listener, what does this mean?
I think it gives you a much deeper appreciation for why sleep feels so essential. It's not just about resting your conscious mind.
It suggests there are really active, dynamic energy processes happening within the very structure of your brain while you sleep.
Exactly. Potentially, your brain's insulation is busy recharging itself, getting ready for the demands of the next day. Sleep isn't passive. It's a crucial period for energy replenishment at this very fundamental structural level.
So we definitely encourage you to think about these ideas. Maybe look into the research yourself if you're curious. What other questions does this raise for you about how sleep works, how your brain functions?
And maybe one final thought to leave you with. This model puts a lot of emphasis on glial cells, the cells that make myelin in brain energy management during sleep.
Right, shifting focus a bit away from just the neurons.
Yeah, and that fits interestingly with some other recent findings, like in fruit flies, where sleep deprivation seemed to affect glial metabolism more than neuronal mitochondria, or other suggestions that mitochondria might not be the absolute central regulators of sleep need.
So this myelin capacitor model offers a glial-centric perspective that might help make sense of some of those other observations.
It potentially could, yes. It suggests the energy story of sleep might be more distributed, involving this interplay between neurons and their supporting glial cells, particularly myelin, in ways we hadn't fully appreciated. It's a complex picture.
Absolutely fascinating stuff. Please don't forget to like the video and subscribe if you want more content like this.